Thanks, friends. We are entering part 2 of this unit today. So I like to call this unit metabolism. AP, I believe, calls it cellular energetics. But there's 3 parts in this unit. So the first part's enzymes. So we wrapped that up yesterday. Hopefully the toothpick ace lab was a good way to kind of demonstrate what happens when you change different environmental effects. So just to revisit a little bit from last class, in your toothpick ace, you should have seen when you change the concentration of the enzyme or change the concentration of the substrate, then that changed the concentration of the reaction. So the enzyme here was you. Okay, you were responsible for the chemical reaction. The substrate that you were working on were the toothpicks. Okay, and so if you had more people breaking the toothpicks, then you could increase your reaction rate, right? But only to a point, only to the point where you could, break them as fast as you were getting them, as many toothpicks as you had. There's a limit as to how fast you can break toothpicks. We're only physically capable of so much. And the same is true for the enzymes. If you increase the concentration of the substrate, it also changes the rate of the reaction. So let's say that you can break a toothpick every second, okay? But let's say you're not given a toothpick every second. That's going to slow down your reaction rate if the substrate is not available, okay? But if you have enough substrate, then that's going to speed up your reaction rate up to a point, okay? Up to the point that the substrate is there's more substrate than what you can physically do, than what the enzyme, if all the enzymes being utilized by the substrate. Or in other words, if every person has a toothpick in their hand, we can't speed that up really any faster, assuming they're going as fast as possible. Now, one of the simulations was for you to dip your hands in cold water, okay? And so if you dip your hands in cold water, that lowered the temperature, and your hands were probably cold, and you're suffering from temporary hypothermia here, and so it doesn't work as fast. So what we should see in that, and what we know about chemistry and molecules, is that when you increase temperature is a measure of kinetic energy. So when you increase the temperature, you increase the kinetic energy. And so those molecules, kinetic energy is the energy of motion, those molecules move faster. Right? And so if they move faster, then it's more likely that the enzyme and the substrate will come in contact with one another. Okay? If they're spread very far apart, they can't interact. But if we increase the temperature and make them move faster and they have more collisions with one another, then they can bind and cause a reaction. Okay? Now, you might also, if you have an inhibitor. Now, I don't think this was simulated in yesterday's activity. But if you had an inhibitor. So let's say on your plate of toothpicks, you not only had toothpicks, but you also had rubber bands, okay? And so you would, reach for whatever you could get a hold of without looking, and sometimes you might grab a toothpick. Well, you're a toothpick ace, so that's what you're good at breaking apart, okay? And so you can break that apart. Well, sometimes you might grab the rubber band, but toothpick ace doesn't break down rubber bands. You need something else. You need rubber band ace to break down rubber bands, which might be like scissors or something, because rubber bands would be hard to break with your hands. And so that would be an example of an inhibitor. That's going to slow down the rate of the reaction because the rubber bands get in the way of the toothpicks and of them being able to do their job, or of them being able to bind with the toothpick ace to facilitate that reaction. So that's just a little bit of a rundown from yesterday's activity. That would be a competitive inhibitor in the example of the toothpick ACE, because it's binding there on the active site, which is in this simulation, your hands, your fingers, where the reaction happens. Okay. Now, a non-competitive inhibitor would be like if I put on gloves. Okay. If I had you put on mittens. So it doesn't really change the shape of the active. I mean, it does to some degree. It changes the shape of the active site and makes it less efficient as doing its job. If you put on mittens, I can imagine that wearing mittens would make it harder to be able to break down these toothpicks, and so that would be a non-competitive inhibitor. Okay, somewhat. So let's go ahead and get started with our lesson today. So just warming up, the difference between competitive inhibition and non-competitive inhibition is whether or not they bind to the active site. Okay, competitive is going to bind to the active site and block the enzyme from getting there. Non-competitive is going to bind to somewhere else on the enzyme, but still cause a change in shape that makes the process less effective. Okay? And then describe the negative feedback demonstrated by ATP and ADP. And so what this is, we didn't talk about it specifically with ATP and ADP, but they demonstrate negative feedback very similar to the way that we discussed in this diagram, where it causes its own production. If there's phosphate available, ATP will continue to be made. If there's no phosphate available, ATP will stop being made. Okay, phosphates and ADPs. And remember, ATP is cycled, it's recycled. It's not really used up. The parts are just broken down to be later put back together. Okay? So let's jump in to this second part of the unit today, cellular respiration. And so cellular respiration is the second part, and then photosynthesis, like I said, are the third part. But we're going to talk about them a little bit together here today. So cellular respiration, the equation for that is C6H12O6 plus 6O2, I'm writing the balanced equation here, yields 6CO2 plus 6H2O. Okay, that's the balanced equation for cellular respiration. Sometimes you'll see it without the sixes written, and you remember balancing equations from chemistry, so it's just the balanced or the unbalanced form. Now, the equation for photosynthesis is just opposite of that. So it's 6CO2 plus 6H2O yields C6H12O6 plus 6O2. Okay, so you'll want to make sure to write those down. Those are very important. And so you see the reactants in photosynthesis are the products in cellular respiration, and vice versa. The reactants in cellular respiration are the products in photosynthesis. Okay? Now, one other thing I want to point out. In cellular respiration, while it's not technically a product, I mean, it is a product. Energy is the product. But energy is on the right side of the equation. It's a product. Okay? We don't write it in the equation, but it is a product. Versus in photosynthesis, energy is a reactant. It's on the left side of the equation. So, in photosynthesis, that energy comes from the sun. And we see that here in this diagram. So, photosynthesis happens in the chloroplasts. Okay? The sun provides that energy for that process. Okay? And you see here the reactants, CO2 plus H2O just like I told you. And the products, organic molecules, which is really C6H12O6 and O2. Okay? And then you can see here how this is kind of a cycle. Those organic molecules, or the glucose and O2, are the reactants which are put into cellular respiration, which happens in the mitochondria. That's really important. Got to know the locations of these. And the products that are produced are this CO2 and H2O. Okay? And notice that the energy is released. It's coming out in the form of ATP. Okay? And is also shown is released as heat. Okay? So it's ATP, which provides work, energy for work, and that work causes heat to be produced. So, for example, in your body, you get glucose from your food, right? You break down glucose in the process of cellular respiration to make ATP, because your cells can't use glucose as energy. They need ATP as energy. And so the ATP provides the energy for cellular transport, for the movement of the, on the microfilaments of those motor proteins. For everything in the body that needs energy, ATP is the energy form. Well, all of these reactions, the sum of these reactions, the sum of all the chemical reactions in the body is known as metabolism. And chemical reactions, as they occur, produce heat. Okay? In chemistry, you learned that you have physical changes and chemical changes. And oftentimes when you have a chemical change, such as a chemical reaction, heat is a byproduct of that change. Okay? And so the heat is produced. And that's why you maintain a body temperature of 98.6, because all of the chemical reactions that normally happen in your body on a daily basis release heat. And the sum of those reactions, the sum of the heat that makes is approximately 98.6 degrees. Okay, and we talked about enzymes. And so for most of the enzymes in your body, that's the optimal temperature in which they work, 98.6 degrees. If you get them out of that temperature range, they don't work as well. And that's why you feel like crap when you're running a fever, because your body isn't performing at its optimal level. Okay, so we would call, because energy is released, this would be an exergonic reaction. Or you might call it an exothermic reaction, because heat is released. So energy is really the exergonic, heat is the exothermic. Versus over here in photosynthesis, that would be an endergonic or an endothermic reaction. Okay? So, that's a lot of information, but that's really important. Okay? Glucose provides the main fuel for our body. And glucose is a sugar. It ends in ose. It's a sugar. It comes from carbohydrates. Now, we learned in biochemistry is going to come back here a lot. We learned in biochemistry that glucose is good for energy, but lipids are also good for energy. Okay? But glucose is really the preferential fuel that your body uses. If it has glucose available, that's what it's going to use. Okay? And so some of you may be familiar with like a low carb diet or a keto diet that's kind of popular right now. And so people that are on a ketogenic diet, they starve themselves of glucose. They do not give their body carbs, which contain sugars, which contains glucose. Even natural sugars that are found in like a lot of fruits and even some vegetables like potatoes and stuff have a lot of natural sugars in them. People that are on a ketogenic diet, they don't eat glucose. And so their body has to switch to another form of energy and use that. And so the second choice then is fat. And fat's lipids, right? Fats and oils or lipids. And so then they'll burn stored fat in their body because they're not feeding themselves any glucose basically on a daily basis. And so their body will switch to burn lipids, to burn stored fat. And lipids are a great source of energy too. Okay, and we'll talk about why glucose and lipids are both, sugars and lipids are both really good energy storage molecules. Okay, and so that's how that works. So, when we look at cellular respiration as a process, it can occur in two different ways. So it can happen aerobically. You should probably be able to figure out what that means by the name. Or anaerobically. Aerobic means that oxygen is present. Anaerobic, oxygen is not present. And anaerobic respiration is also known as fermentation. Okay, remember, oxygen was one of our reactants. So most of the time, hopefully, cellular respiration is going to proceed aerobically. This is going to make much more ATP. Okay, sometimes though, cellular respiration will proceed anaerobically, but this will make less ATP. Okay, we're going to spend most of our time talking about aerobic respiration, but on the last day of our cellular respiration portion of the lesson, we will talk about anaerobic respiration or fermentation. But basically what I want to tell you, just as an overview about them today, is that most of the time we have plenty of oxygen, right? We have all the oxygen that we need. I don't know that I've ever struggled to breathe, except when I'm exercising. And you've probably experienced that too. Y'all are in better shape than me, most likely. But when you exercise, your body does not have the oxygen necessarily that it needs to do aerobic cellular respiration efficiently or effectively. And so, obviously, when you're exercising, your body needs lots of ATP. And so, and I mean exercise like aerobic exercises, like running, like playing basketball, not things like, well, they're still exercising great for you, yoga or, I don't know, so, light swimming, something like that. I'm talking about more of a heavy aerobic exercise where you increase your breathing significantly. So, when you're exercising hard, though, you do not have as much oxygen as you need. But your body still needs ATP to carry out those processes. And so when I say more and less, I'm talking about relevant numbers like 36 and 2. And you don't have to know those now, but just to give you a little perspective, it's a lot more and it's a lot less when there's with or without oxygen. But your body still needs ATP to do its things when you're exercising. But oxygen is not available to carry out cellular respiration in its natural form. And so it reverts to anaerobic cellular respiration or fermentation, which allows it to still make some ATP. Okay, and we'll talk about how that happens and the pathways more later. So, as we talk about photosynthesis and cellular respiration, photosynthesis and cellular respiration are a series of redox reactions. Ideally, we would have gotten to this in biochemistry last year, but because of COVID, we just didn't get that far. So I apologize for that, but that's okay. We're going to cover all that we need to know about redox reactions right here, right now. So redox stands for reduction and oxidation, okay? Oxidation and reduction. So loss of electrons is referred to as oxidation. Gain of electrons is referred to as reduction. Now, this is a little bit counterintuitive. You would think reduced means loss, but that's not true. Gain is reduction. And so in chemistry, we usually teach two different mnemonics that kind of help students to remember this thing. So Leo, the lion, says Gur. So that means loss equals oxidation, gain equals reduction. Leo, the lion, says Gur. Okay, loss equals oxidation, gain equals reduction. Or you may also think of oil rig. Oxidation is loss, reduction is gain. And again, what we're gaining or losing is these electrons. So remember, cellular respiration and photosynthesis are chemical reactions, right? And what you should remember from biochemistry is that in a chemical reaction, basically what we do is we break elements apart and we rebuild something new with them. It's kind of like Legos. It's kind of like if you have a Lego set and it's supposed to make, you can make two different things with your Lego set. Or you can break those things apart and use the same pieces to build new things. And that's what happens in a chemical reaction. We are given carbon and hydrogen and oxygen and, more oxygen over here. And these represent three different Lego pieces. And we could build glucose and oxygen with them. Or we could use them and build carbon dioxide and water. And it still uses all of our pieces. That's a balanced chemical equation when it still uses all of your pieces. And so when we break down these molecules, we are breaking bonds apart. Okay? Now, what are bonds? In chemistry, and I think in biochemistry, we talked about this. We've got a single bond, a double bond, and a triple bond here. Okay? Well, if you remember, bonds are made up of electrons. So in a single bond, there's 2 electrons. In a double bond, there's 4 valence electrons. All of them are valence electrons. And then in a triple bond, there's 6 electrons. Valence electrons are the ones that are involved in bonding. And so, remember in chemistry, you drew the Lewis dot structures, and those will be the ones that were involved in bonding. Those valence electrons made that double bond that you see here with those 4 dots. Okay, taking you back to chemistry just a little there. So we're not going to really focus about the double and the triple bonds today, because most of what we are talking about here are single bonds, specifically with glucose. But it has 2 electrons. So when you break that bond apart, you release those electrons, okay? And an element either loses electrons in the process, or not an element, a compound or a molecule can lose electrons in this process, or it can gain electrons in this process. So when a bond is broken, an element that has lost electrons is said to be oxidized. An molecule or compound that gains electrons is said to be reduced. Okay? And that's really important. So we're going to stop there with oxidation and reduction and talk about kind of 2 other things here. So why are carbs and fats the best molecules for energy? You should say because of the bonds, okay? Because of the number of bonds that are present. If you look at the structure of a lipid, it's a long carbon chain with hydrogens. I'm running out of space, so I'm not going to draw the H's, but there should be an H on this other end of every one of these, all the way around. That fatty acid chain, okay? Remember, it can be saturated or unsaturated. If it had a double bond in it, that would make it unsaturated. If it's all single bonds, that would make it saturated. But there's lots of bonds there. And so those bonds represent potential energy. Okay, when you break a bond, not only do you release 2 electrons, but you also release energy. And I'm writing this down. I want you to write it down too, because that's going to be important later. Okay, every time we break a bond, we're going to release just a little bit of stored potential energy. So lipids have lots of those bonds that can be broken down. The same is true for carbohydrates and glucose. They have lots of bonds easily broken down to release energy. Okay? Now, the second part of this that I want to talk about. Why must glucose be broken down in a series of steps rather than in one quick reaction? Okay? So, cellular respiration, as I've mentioned, is the process of breaking down glucose and making energy out of it. Okay? We need oxygen to do it, and it's going to make some other stuff in the process. But the goal of cellular respiration is to break down glucose and make energy that the cells can use. Cells can't use glucose. Cells can use ATP. Cellular respiration is the process that turns glucose into ATP. Okay? So, if you broke down cellular respiration, proceeds in 4 steps. We'll talk about those tomorrow. There's 4 steps of cellular respiration. Okay? And each of those steps really has a lot more smaller steps in the process. Okay? So it's like 4 steps with like multi-steps in each step. But it's broken down in a series of many steps or a series of many redox reactions. And the reason for that is to be able to break it apart little by little. Okay? If you took a stick of dynamite, that represents a lot of potential energy, right? And we could release all of the energy in that dynamite in one reaction. If we could just break it all apart at once, we would just have a huge explosion of energy, right? And that's what you do when you have a stick of dynamite. You light the end and it all explodes at once, okay? But the problem is the body or the organism that this is happening in can't capture all of that energy at once. It's just too much to do anything with, okay? We need a little amount. We need small amounts of continual energy, not these gigantic explosions of energy. And so, what if instead we could slice that dynamite into tiny slices and use it as needed? Okay, set them off individually as needed. And then we could harness the energy better, or we could better capture that energy. And so that's kind of what's being done with glucose. Glucose has a lot of energy. But if we break it all down in one big fail swoop, we can't harness it. We can't capture it. It's too much to use. Okay? So instead, glucose is broken down in a series of steps. All right. So, one of the steps in cellular respiration, I'm not expecting you to get all of these steps today, but I'm going to write them out. There's 4, like I said. The last step of cellular respiration is what's called the electron transport chain. Okay? And that's where most of the energy comes from. In cellular respiration, there are about 36 ATPs total that are made. Okay? But about 28 of those ATPs come from the electron transport chain. So it is, that's where most of the energy happens at. All right? And that's going to be important in just a minute. So, because this is a series of oxidation and reduction reactions, we have these electrons. Remember that things are constantly losing electrons and gaining electrons. We have electrons that need a little guidance, okay? And so we have molecules that are known as electron carriers. So NAD, which stands for nicotinamide adenine dinucleotide, is an electron carrier. When it gains electrons, it becomes NADH, okay? So this is no electrons or without electrons, and this is after it gained electrons, okay? NAD becomes NADH. And so when in this process of cellular respiration, as in these steps, bonds are broken and electrons are released, okay? And so as those electrons are released, NAD plus picks them up and it carries them to the electron transport chain, okay? And in the electron transport chain, these electrons, because it's their transport chain, that's where they have to go, they are used to make energy, to make all of those 28 ATPs that we just mentioned, okay? So we have enzymes, because this is a chemical reaction, that help to catalyze these changes in these processes. And so one of those enzymes is dehydrogenase, okay? And so dehydrogenase, it dehydrogen, when hydrogens are removed, when hydrogens are broken off, remember these were our bonds there that contain hydrogens. When those bonds are broken apart, electrons are released. And so it helps pick up those electrons from glucose or another substrate like a lipid and transfers them to NAD. Okay, so dehydrogenase is involved in carrying these in transferring the electrons to NAD, which forms NADH and takes them to the electron transport chain. Okay, NAD picks up electrons and becomes NADH. Dehydrogenase is just an enzyme that allows it to do that. Okay. So I want you to think about NAD and NADH similar to the way that you think about ADP and ATP. Remember, ADP picks up a phosphate and it becomes ATP. ATP is really the energy-rich molecule, okay? And then it can be broken back down and recycled back into ADP, okay? Once that other phosphate is broken off. NAD and NADH are very, very similar, okay? Except instead of picking up a phosphate, they're picking up electrons. When it picks up electrons, it becomes NADH, okay? That's more of the potential energy molecule, NADH. And NADH carries those electrons, again, to the electron transport chain. Once it drops them off at the electron transport chain, it goes back to being NAD until it can pick up more electrons and carry them to the electron transport chain. So this is an electron carrier because that's what it does. It carries electrons, okay? This is just an image. Here's our NAD molecule, okay? This is the oxidized form, meaning it's lost electrons. This is the reduced form. So after it gains these electrons, also gains a hydrogen, it becomes reduced. See, there's that extra hydrogen that it gained. And those electrons right here that made that bond that allowed that hydrogen to be stuck on there. You don't see the hydrogen over here in the oxidized form. Everything else is the same except that hydrogen plus those two electrons that you needed to add it there. Okay? So here was our enzyme in the middle, dehydrogenase, that facilitates that process. Okay? Now, this can go back to this form. If it loses those electrons, if it's oxidized and loses those electrons, then it will revert back to NAD. Just like ATP, NAD and NADH is cycled or recycled. Okay? So, as we said, glucose is broken down in many small reactions, and the electrons are shuttled by NADH down the electron transport chain, also known as ETC. Okay? Now, ultimately, oxygen is the final electron acceptor. What does that mean? Well, it's a concept that's really important in cellular respiration. You used to frequently see AP tests that said, what's the final electron acceptor? And oxygen. Okay, oxygen's the final electron acceptor. But what does that mean? That's not usually a test question on current AP tests because they've kind of redone the way that they ask questions. But you might see it on one of your AP tests. So, what does that mean? Well, if you remember, oxygen was one of our Oxygen, bear with me. I erased all my writing. Oxygen was one of our reactants in cellular respiration. Remember, we had C6H12O6 plus O2 yields CO2 plus H2O, and this isn't balanced, but that's okay. But oxygen's one of our reactants, meaning it really needs to happen. You have to have oxygen for the electron transport chain. Well, what do you know about oxygen? You should say oxygen is one of those FON elements, which are very electronegative. Going back to biochemistry again. They are electronegative. F, O, and N are the three most electronegative elements. Now, what does that mean to be electronegative? It means that they hoard electrons. They have a strong affinity for electrons. They pull electrons towards them. Okay? And so, in that fourth step of cellular respiration, the electron transport chain, we have to have a reason to get electrons there. And it's because of oxygen. The electrons are shuttled to where oxygen is. And once NADH releases those electrons, they go to oxygen. Okay? And when these oxygen pick up the electrons, guess what they make? They make water, one of our products of cellular respiration. So, oxygen picks up those electrons from NADH in the electron transport chain, and these electrons then are used to drive the synthesis of ATP. Remember, in the electron transport chain, we said there were 28 ATP molecules? Okay. Those come from these NADHs, right? Those electrons that NADH picked up, these electrons are really responsible for making the ATPs, okay? The goal of cellular respiration is to break down glucose in lots of little steps, grab those electrons, and take them to the electron transport chain where they can most efficiently make ATP, okay? And at the electron transport chain, they're drawn there because of oxygen, because oxygen is electronegative. Once oxygen accepts those electrons, it will actually convert them into water with other available hydrogen. And that's one of the byproducts of cellular respiration. So, I know that was a lot. Okay? It's going to get easier, I promise. We're going to draw lots of diagrams as we go through this. So in the back of your binder packet that you picked up last week, you have lots of white pieces of paper. So when you come next time for the lesson tomorrow, bring one of those white sheets of paper with you and a pencil, because we are going to draw out a bunch of these diagrams, okay? And every day, as we build on it, you'll understand it better, okay? So today we were just kind of hitting the highlights. I want you to understand NAD and how it converts to NADH and how it's used as an electron carrier. For all things good, you got to know this reaction, okay? And I really want you to have a good grasp on this as well, okay? That these processes are reversible, where they happen in the chloroplast and in the mitochondria, that energy is released in one and used in the other, and that energy can be converted into heat. You also need to understand the difference in aerobic and anaerobic respiration. Basically, at this point, just understanding it's with or without oxygen. And then finally, I want you to really understand oxidation and reduction, and that those are loss and gains. So this was a lot of information. This is really the least important part of it. Okay? Everything else that we talked about today is truly really important. Okay? If you forget anything, forget about dehydrogenase. But everything today is really, really important. I didn't give you any other activities today because I want you to pause and try to absorb this, because it was a long lesson and I'm sorry you had to listen to me for so long. But it's really important, and you got to nail it before you can move on tomorrow. So with that, you all have a great day. If you have any questions, let me know. And if not, you'll hear from me again tomorrow. See ya.